Check, check. 1, 2.
You're gonna have to refresh that.
We're good to go?
Yeah.
Okay, excellent. Yeah, I just checked it works.
Great.
All right, I think we're gonna get started, everybody. We're having a little audio issue, but I think we have resolved the issue, so we're gonna go ahead and get started. Thank you all for coming. I know it's sort of been a busy week. People have been traveling in and out for ACR, so we appreciate you participating, and those of you online, if you can hear me. It's really my great pleasure to welcome Dr. Rameen Beroukhim here today. He's going to be giving a seminar as part of a joint GSI and PhD effort here. Dr. Beroukhim is an associate professor of medicine at Dana-Farber Cancer Institute. He's also a clinical oncologist, seeing patients as well. And it's funny, in sort of looking over his CV, I was just commenting on the number of pages. He's had a number of really seminal contributions. His lab and research is focused on how chromosome alterations contribute to the development of cancer. He has a specific interest in neuro-oncology. And so for those in PhD who are very neuro-focused, I forced you to listen to some cancer, but at least it will be in the brain, which will be helpful. Yeah, I'm very excited to have him here today and to welcome him for the seminar. So thank you again for coming.
Yeah, thank you very much. I guess I have to turn this on. Yeah, can you all hear me? Yeah. Okay. I want to thank you for inviting me, and it's wonderful to come here. We have so many ties with PU. Actually, my wife is on the faculty here, now adjunct, but so she comes here to treat folks on every other Friday, and so many great scientists here that we work with. So I'm very excited to present. Please do interrupt me if you have any questions. My wife, again, does tell me that when I talk for a long time, people tend to start to get physically ill. And so, if you feel that, just sort of stop, and then you could ask a question that can make people feel a bit better. And I guess we are close to hospitals, so that's good too. Great. So I'm going to talk today about positive and negative selection in cancer genomes. And first to my conflicts, there are those. So when we think about the cancer genome, right? Well, how do cancers form? Well, cancers form due to mutations that they get, throughout their genome. And some of those are oncogenes and mutations in oncogenes and tumor suppressors, but some are not. So actually, the process is that you have cells in your body, and every time they divide, they undergo mutations at random throughout their genomes. Most of those mutations are going to be selectively neutral, as shown in yellow, or even in negatively selected mutations, that's shown in blue. But then when occasionally one cancer or one cell might undergo. Sorry, I'm just going to try to find. Yeah, so one cell might undergo a set of mutations that are positively selected in red here, and that cell then transforms and expands into this tumor with many billions of cells, and each of those cells will have those mutations that cause the transformation, but they'll also have all the other mutations that occurred beforehand that did not cause the transformation, right? And then the cells, as they're dividing, they get more mutations and maybe more, of these driver positively selected mutations as well as more neutral ones, right? So, when we want to understand how cancers form and what's causing cancer, we want to understand these mutations that are transforming, but we can't really tell which of those are transforming just from the genetic profile of the final tumor, because it has all these passenger events that occur alongside, right? So much of cancer genomics has been dedicated to trying to distinguish the red driver events from the blue and yellow passenger events, right? And we actually contributed to that early on, where we developed one of the first methods to look at this, called GSTIC, where, well, I should say, just going back for a second, from a single tumor, you cannot distinguish which are the important mutations, but every cancer has to have had driver mutations. So some mutations that are red like that, not necessarily the same ones, but some limited set. There's some limited set of mutations that can cause cancer, right? Whereas the blue and yellow mutations, they don't have to occur in every cancer, right? So across cancers, you'll tend to see the red ones frequently, and you won't see the blue and yellow ones as frequently. So an approach to detecting these is to use statistics, and so this is an example of a method that we've developed to do that, where we take a bunch of different cancers, and at each locus in the genome, we ask, well, is there a copy number change? Did you gain copies or lose copies at each of these loci? And then we add up the total amount of copy change over all those tumors at each of these loci. And then we ask, well, what would these genomes have looked like? So this profile, we don't know which of these is likely to, is positively selected. But we asked, well, what would these genomes have looked like if these were passenger mutations that could have happened anywhere in the genome with equal frequency, right? That they weren't selecting a particular gene, they just happened randomly. And to model that, we permute the locations of every one of these alterations and then reform these sums, right? And so you can get, here this is the G-score representing the sum of mutations. You can get a distribution of sums that you'd expect by chance. And you can say, well, anything past this green line would have been unlikely to happen by chance. And so you can apply that threshold to detect driver copy number alterations. And so this method, we developed almost 20 years ago now, and it's been used a lot across many different cancer genome studies. And we've discovered a lot of things. So this is an example actually from 12 years ago, where we found 70 regions that were frequently significantly deleted, 70 regions of significant amplification. And so these amplified regions tend to contain known oncogenes like LMYC, MCL1, MDM4, FGFR, EGFR, and so on, as well as some regions that don't contain known oncogenes, which we think are areas for discovery. And then these regions often contain tumor suppressor genes like NF1 and P10, P16, and so on, although they also contain some fragile sites. And so, this was successful in identifying oncogenes and tumor suppressor genes. Actually, MCL1, I think partly based upon our studies, Novartis decided that they wanted to develop MCL1 inhibitors, thinking it's an oncogene in cancer. And so those are, I think, being tested out in the clinic right now. And you look at many of these, they're CDK4, MDM2, EGFR, PDGFRA, they all have inhibitors against them that are, in many cases used in the clinic. So it can be a way of identifying therapeutic targets, right? So that we did that for copy number alterations, and then actually recently there were other methods developed for mutations that do much the same thing, where they take you take profiles of every tumor and you add up the mutations across all those tumors, and you get identify loci that are mutated more often than you'd expect based upon some significance threshold, and those are the likely driver loci, right? And again, based upon this sort of analysis, you can identify, in this case, 224 cancer genes, right? So there's been a ton of effort to try to identify positive selection in cancer genomes. But I mentioned that I had three colors there. There was the positive selection in red, the neutral in yellow, and then negative selection in blue, right? So what about negative selection? Is there negative selection going on in cancer genomes? And would we even care if there was, right? Well, in fact, not very many people studied negative selection. And one of the reasons why is that in the point mutation world, there isn't much. So there are a couple of groups that came up with this, similar results in 2017. The Peter Campbell's group, led by Inigo Martin Carena, and then Shamil Sunayev's group. And these figures are from the Martin Carena paper, where they took the mutations in every gene and asked, do they tend to be more often synonymous, DS, or non-synonymous, DN, right? So if there's a certain rate at which mutations would be expected to be non-synonymous and if they occur at random. And so you expect sort of 3.5 of these non-synonymous ones to every one of these synonymous ones. And they asked, well, the ratio that we observe, is it in accord with that? So that would be like, if it's the ratio that expect, it would be 1 here. And across cancers, they found actually that most often the nonsense and missense mutations exceeded what you'd expect from this ratio, right? And so you would expect that if these mutations, nonsense and missense mutations, were negatively selected, they would tend to be purified away, and so you'd see fewer non-synonymous mutations compared to what you'd expect. But in fact, they saw more than you'd expect, suggesting that the major selective pressure is actually positive, that cancers are trying to, are mostly identifying, that more mutations cause a positive effect for cancer transformation and growth than there are, that cause negative effects. And so that's sort of quantified here, where about almost 2% of genes undergo mutations that seem to undergo positive selection in cancer, whereas about less than 1% of genes undergo what apparent negative selection, right? And that's actually absolutely the opposite of what I would have expected, right? Because there are a lot of ways to break a cell, and but here it's saying that there are more ways to make it sort of power it up than to break it, which is surprising to me. And one reason for it is that you have 2 copies of every gene, and so many of these mutations that would have been under negative selection are protected by the second copy; they're buffered. But nevertheless, the fact is that there's not much negative selection in mutations in cancer, point mutations, right? So is that it? Are we done? Well, in fact, point mutations aren't the be-all and end-all in cancer. If you look at the average cancer genome, about 1 in a million bases is mutated, right? Whereas 1 in about 3 bases is altered by copy number alterations, gains or losses of copies of DNA, right? So having 5 copies of a gene or 0 copies of a gene, right? So 1/3 of the cancer genome is affected by these, 1 in a million bases by these, right? So if you know anybody who works on point mutations in cancer, their lives have been wasted, right? Because they're looking at that. We look at the copy number alterations, right? So not much negative selection in point mutations, but copy number alterations are what's important. What about there? Well, actually, I should say, not only are the copy number alterations affect more of the genome than any other type of genetic alteration, including point mutations, they are the most frequent genetic alterations in cancer. So this is a view of the most frequent genetic alterations in cancer, ranked by the first to the last. Well, this isn't the last; it's still pretty frequent. But the most common event occurring in almost 40% of tumors in the Cancer Genome Atlas is mutations of p53. So that is a mutation. But actually, those mutations tend to get you copy number alterations because p53 is the guardian of the genome. And the number 2 most significant, most frequent event is amplifications of 20q, gains of the entire chromosome arm, 20q. The second is gains of the entire chromosome of 7p and so on. So actually, among the 25 most frequent events in cancer, this is the 25th most frequent. It's deletions of the gene p16. This is a focal event that tends to only affect a few genes. But these 23 actually are, so these 24 are all copy number alterations, and 23 of them are copy number alterations that encompass the entire chromosome arm on which they lie, right? So that's why so much of the genome is affected by copy number alterations, because you have massive amounts of the genome that are affected by aneuploidies, alterations of whole chromosome arms or whole chromosomes. So that's about almost a quarter of the genome is affected by these arm-level events, right, in cancer. So, there are all these arm-level events that are occurring massively frequently, and each of them is affecting like hundreds to 1,000 genes or more, so affecting huge amounts of the genome. And we actually don't know what they do. So they were the first events to be discovered in cancer. Boveri, sort of came up with a genetic hypothesis of cancer based upon seeing aneuploidies, right? But we looked to see, which of these alterations affects a known cancer driver, and likely is targeting a known cancer driver. And we did that with some pretty loose criteria, where we said that, well, if 1 out of every 5 times that you see, a 20q amplification or a 7q amplification or whatever, you also see a focal event altering a known oncogene or tumor suppressor gene, then we would say we can explain that event, that arm-level event, right? And all of these, and that doesn't even go into the 80% where you don't see the focal event, right? So even among those, we think we don't necessarily understand what's going on, because again, they're very complex beasts. They affect so many genes. But even with those sort of loose definition criteria, all these things in stars have no known driver events. We don't know why they're so frequently amplified and deleted. So I see this actually as the most important question in cancer genomics, in the cancer genome, cancer genetics, is what are these aneuploidies doing, the most frequent events in the genome, the ones that have the most impact. Going from the earlier question, well, does negative selection matter, and does it exist? Well, so we can ask, do these copy number alterations undergo negative selection, and should we care? And I'll say that there are actually 2 reasons to care. One is that, well, negative selection is indicating things that cancers don't like. If you inactivate a gene, if alterations that inactivate a gene are negatively selected in cancer, that means the cancers are trying to avoid inactivating that gene. So that means that if you have a treatment that inactivates that gene, the cancer won't like it, right? So that seems like a reasonable approach to look for treatments, and so that, a way to find therapeutic opportunities. But even more than that, you actually, for copy number alterations, you need to understand negative selection to understand positive selection. Why do I say that? Well, this is an example of a locus that undergoes frequent copy number alteration. Actually, it's the most frequently deleted region in the genome, covering P16, CDKN2A. It's on 9p, right? And so each of these columns is a different cancer, this is just a region of the genome with a P terminus up here and the centromere down below. And P16 is here, and these regions in blue are deletions. Dark blue is homozygous deletion, light blue is hemizygous loss, and then regions in white have no copy number change. And you can see that, P16, there's a ton of homozygous deletions that cover P16. That makes sense because it's a tumor suppressor gene. It's one of the most important tumor suppressor genes in the genome. We know that because it's the most frequently deleted tumor suppressor gene in the genome. But then if you look at these deletions, they can extend over quite a distance, right? In fact, they extend over like megabases. So, and some of them go right up here. This is the largest deletion that we see in this direction. It goes up here, but doesn't go any further. And several of them get to that sort of near that point, right? Why does it go further? Well, actually, like when I was doing a different project that's sort of unrelated to the one I'm going to discuss now, we were looking at different essential genes in the genome, and it turns out that SMU1 is involved in RNA processing. It's a gene that's essential for cells to survive. Cells can't tolerate its loss, right? So you could imagine that if a deletion of P16 extended across SMU1, the cancer would die, right? So there are no deletions that cover this because of negative selection against deletions of that locus, right? Now, what if SMU1 had been closer to P16? Then the universe of deletions that are available to a cell that would take out P16 and not kill the cell by taking out SMU1 at the same time, the universe of those deletions would be smaller, right? And so you would see fewer deletions of P16 and fewer cancers, right? So the reason we think P16 is such an important tumor suppressor gene is because SMU1 is as far away from it as it is. To understand positive selection in copy number space, you have to understand negative selection. Right, so what's another example of that? NF1. NF1 is a potent tumor suppressor gene. Like there are kids who are born with mutations in NF1 who get congenital tumors. I had a friend, Karen Chakowski, who came up to me many years ago and said, we're doing studies on NF1. We think that it's an important tumor suppressor gene in sporadic glioblastomas. And I know that you're looking at copy number changes. Do you see deletions of NF1? And I looked, and there were a few deletions of NF1, but it did not attain statistical significance from our gistic methods that I described before, right? So I said, well, there's some deletions, but it's not statistically significant, so I don't think it's actually driving. And she heard there's some deletions. She says, great. So you have evidence that it's a tumor suppressor gene. And I was like, so she published a paper, and that's a tumor suppressor gene in glioblastoma. And she was right, and I was wrong. NF1 is absolutely a tumor suppressor gene in glioblastoma, right? It's also has point mutations and all that. Why doesn't it undergo deletions? Well, UTP6 is an essential gene that's just south of it, and PSMD11 is an essential gene that's just north, but megabase on this side and half a megabase on that side. So there are very few deletions that can take out NF1 that don't also take out an essential gene. So that's why you don't see it deleted so frequently, right? And so we did not detect it because of the negative selection. When we did a pan-cancer analysis of the most frequent, most significant deletions across cancers, which are likely to contain tumor suppressor genes, the most significant site was P16. There are a lot of these fragile sites which tend to get deleted due to mechanistic things, which come up in our lists. RB1 is a known tumor suppressor gene. We don't know what's in here. P53 is the most important tumor suppressor gene in the cancer genome, and it came in at number 35, right? So what the hell is it doing at number 35? Why isn't it up at number 2? It's because it's not deleted very frequently. Why is P53 not deleted frequently? Well, P53 is here on chromosome 17p at 7.68 megabases down. RNA Pol IIa is at 7.5, 180 kb away from it, right? RNA Pol IIa, and this is these are depth map plots where the Broad Institute sort of applied CRISPR, knocked out RNA Pol II across. Well, actually, this is RNAi. It applied RNAi to knock it down across many, many different cell lines, and in almost every case, it killed those cells, right? It's an extremely essential gene, right? And only 180 kb away. About 500 kb away on the towards the centromere is aurora kinase B, and this is CRISPR knockout and RNAi. Again, killing these gene effects are heavily negative. They're killing those cells. So absolutely essential genes, right? So cancers don't delete P53 very frequently, probably because, at least in part, because they can't without taking out RNA Pol II or aurora kinase B. And for all I know, these genes are as close to P53 as they are as a way to prevent cancer, right? The conclusion from this is that if you want to detect positive selection, you need to understand negative selection. It also means that we need to consider then, that those P16 deletions, not just what they covered, but what they didn't include, right? Where they stopped. So there's start and end points to identify things like SMU1, right? So, okay, what determines the start and end points of a copy number alteration? It's the rearrangements that generate them. Every copy number alteration in a cancer genome that is within a chromosome is caused by a rearrangement, right? So you can have events where you have gains and losses of whole chromosomes from non-disjunction, right? Those don't require rearrangements. But every interstitial event requires a rearrangement. And so you can see that here. So that we did a whole genome sequencing of this cancer sample, and we asked how much DNA was there at every locus. And so the amount of DNA at each locus reflects the number of copies of DNA that are being sequenced, right? And so we're seeing that there's sort of a constant amount of DNA, which is reflects 2 copies, right? And then here, all of a sudden, there's a drop that there's like half as much DNA, about 1 copy, right? And then it jumps up again. So this is a deletion of this locus, right? And it so happens that this is whole genome sequencing data, so we could also look at the rearrangements, and we found bits of DNA that were stuck together that we didn't expect, indicating that this breakpoint was rearranged to this breakpoint. They were connected to each other. And if you think about it, that has to happen, right? If you take a chromosome and you take out a bit of it, you all of a sudden leave these open ends, and they have to attach to something, right? And so in this case, they attach to each other. They could have attached to some other chromosome, but they have to have a rearrangement, right? So what determines the ends of the copy number alterations is the rearrangements that form them, right? What determines the length of the rearrangements? What bounds rearrangements? Well, this is a view of the rearrangements across 2,700 cancers, where the genome is along each of these axes. So actually, all 3 axes, so this down here is chromosome 1. This is chromosome X. So a dot here is going to be a rearrangement between chromosome 1 and chromosome 2. Right, so the different dots are basically indicating which 2 sites in the genome are being connected by each of these rearrangements, right? And you can see, a couple things. One is that some parts of the genome are broken more often than others, and they just connect everywhere, right, like this. But the other is that most rearrangements are within chromosomes, and even within the chromosome, most of them are short, right? And in fact, the frequency with which you see rearrangements decreases as 1 over the distance between their breakpoints, right? What's determining this pattern? The 3D structure of the genome. So this is a high-C map of the 3D structure of the genome, next to that, what we were just looking at, the rearrangements in cancers. And again, you can see that most of these connections, the close regions are sort of close to each other in 1 dimension as well, right? Why would the rearrangements of cancer follow this high-C map? Well, what is the high-C experimental procedure? You take the cells, you fix them with like formalin or something, cross-linking the DNA, and then you sequence across those cross-links. In other words, you generate rearrangements, and then you sequence across them, right, to figure out which bits of DNA were right next to each other. So of course, you'd expect that cancer rearrangements would look like high-C data, because you're generating rearrangements to get the high-C, and the cancers are generating rearrangements, right? So they follow this. So the 3D genome structure predicts what loci connect to each other about 2,600 times better than any other factor. And the effect of that is that the most frequent events are the shortest ones. And then they decrease in frequency as you get longer, except for events that are exactly the length of a chromosome arm or 2 chromosome arms, meaning a whole chromosome, right? And so these are focal copy number alterations, like the deletion I'd showed you earlier. And then these are the arm level events that take up all of 20p or 20q or whatever, right? So that's the length distribution of copy number alterations. Now, we can break it up. As I said, there's the interstitial, the ones that are internal to a chromosome. They go like the 1 over L. But if you think about it, events that start at the telomere and go inward, they don't need to connect anywhere. You could just chop off a telomere and let it float away, and that's it. So so you'd expect that those might have a different length distribution, and in fact, they do. So events that start at the telomere, it's a smile distribution where, like, really, for all intents and purposes, it's the breaks can happen anywhere in the chromosome arm with equal likelihood, and so you get a uniform distribution of lengths of telomeric events. And the same is true for centromeric events, right? If you want to look at the boundaries of copy number of events, you can look at internal ones, which have this actually kind of complex sort of distribution, or you could look at telomere-bounded ones, which are basically uniform. So let's think about the uniform ones, and how can we use that to understand positive and negative selection? So we developed this method, Biscuit, which to do just that, where we look at events that start at the telomere and ask where do they end within the chromosome arm, and you sort of sum it up over thousands of cancers, right? And if there's it's a uniform distribution at equal frequency anywhere within the chromosome arm, then that's we have no evidence that there's positive or negative selection going on anywhere. This is a cumulative distribution of this, which I won't go into detail on. But you can imagine an alternative scenario where you have very few short events, but all of a sudden, as soon as you get past this locus, you have a ton of longer events, right? So that means that the cancer wanted to have events that include this locus, right? So that's indication of positive selection for events that include that locus. Conversely, if you have a lot of short events and then they drop off exactly at this point, so that very few long events, then that indicates negative selection, that the cancer is avoiding having events that cover that locus, right? So here is an example of 9p, where you have very few short events, and then at this locus, it suddenly jumps up and you have a lot of events that are longer, right? What's at that locus? It's P16. Again, one of the most important tumor suppressor genes. This is an example of the negative selection on 8q, where you have, short deletions are quite frequent actually, but as soon as the deletion passes this point, it's very infrequent. We have very few deletions that pass that point. What's there? MYC, which is one of the most important oncogenes in cancer. And not only is it an important oncogene, evidently, even when it's not amplified, cancers don't want to delete it. This is sort of the logic that we use then to develop a method to look for evidence of positive and negative selection across cancer genomes. And so we applied it to 11,000 cancers in TCGA, and we detected 193 regions that appeared to be under selection. So here you have positive selection for either amplifications on the right or deletions on the left, and negative selection for amplifications on the right and deletions on the left. So these are like oncogenes. These are like tumor suppressor genes. These are like essential genes and cell essential genes. And then these are like toxic genes, right? That when you amplify them, they're toxic to the cell. And so we identified 90 regions under positive selection. We actually identified more regions that are apparently under negative selection than positive selection. All of these regions. Were we identifying real genes? Well, when we cross our lists of likely oncogenes and tumor suppressor genes with ones that had been described before, there's a cosmic tier 1 genes where these folks have looked through the literature and said, we think this is an oncogene, this is a tumor suppressor gene, and they've made the list for everybody to see. When we cross that, we find that our list is heavily enriched for those tier 1 genes. But many of the genes that we identify were not altered by focal copy number alterations. They were only altered by, these, I should say, many of them were not previously detected in copy number alteration sort of studies, because they tend not to be affected by short events. They tend to be affected more by these long events than like these chromosome arm level events, which indicates that this sort of approach of looking at the edges of often long events is useful. It can detect new things. 103 under negative selection. We just saw that the positive selection seemed to validate. Negative selection also validated. So here we're looking at mutational evidence for against negative selection. So again, this is this DN-DS ratio that we looked at before from Inigo, Martin, Carina. If you look at regions that we think are under positive selection for deletion, the genes in them tend to prefer to get truncating mutations or splices, truncating mutations or missense mutations they prefer those relative to synonymous mutations more often than you'd expect by chance. There's evidence for positive selection of inactivating events in these genes, right? But in the regions that we think are under negative selection for deletions, we see fewer truncating events than you'd expect given the number of synonymous events. The DNA-DS ratio is less than 1. So you remember that previously folks had not been able to find strong evidence of negative selection in cancer genomes. But when we limit to the regions that we think are important, have negative selection for deletions, we do see that this metric starts showing up. So we think that we're identifying both positive and negative selection. We also are able to measure the amount of positive and negative selection that each of these loci is applying. And so when we add that up, we find that the frequency with which you see whole chromosome arm amplifications in this case, but also deletions, is heavily shaped by the amount of negative selection. So this is a fitness score that reflects the amount of negative selection that we think is going on due to the peaks that we see. And we sort of summed it up for each of these chromosome arms. And you can see that the lower the fitness score, the less frequently these, the lower the fitness score for amplifications, the less frequently these chromosome arms are amplified. So we see that negative selection is definitely shaping cancer genomes. So that sort of is some published data. There we were using really only a small subset of the events. As I mentioned, we were using events that start at the telomere or the centromere. We're not using the internal events. The internal events have more information because they have 2 breakpoints, not just 1. They're very frequent. And also we weren't asking what if you lost 1 copy, 2 copies, or if you gained 1 copy, 2 copies, 3 copies, what's the difference? And so I'm not going to go into detail, but just as a teaser, we're working on methods now to do that sort of thing. We're looking at all events, focal and arm level and telomeric and centromeric, and we ask, is there positive selection or negative selection for homozygous deletions, for hemizygous loss, for no change, low-level amplifications or high-level amplifications, right? So at every copy number level, we can make a calculation. And this is an example here where on chromosome 10q, we see positive selection for homozygous deletions of P10, and intriguingly, negative selection for these regions outside it. So these deletions of P10 are limited, delimited, and we think that there are cancer essential genes on over here, and that we can find by this method, right? There are also regions that appear to not want to change. This chromosome doesn't undergo really much of a and then there's some regions that appear to undergo hemizygous loss. Although that might be an effect of the loss of P10, we have to take that into account and then see what the effects are. And we don't see much positive selection for amplifications. We do see a bit of negative selection, which could represent the presence of toxic genes on chromosome 10q. So we're working on this method. My postdoc is working hard on the actual guts of the method with actually a really great undergraduate. And while he's doing that, I'm doing the more important thing, which is trying to think about what the name should be. So if anyone of you have a good name for the method, Biscuit 2.0 after the prior one, or Triscuit, we had Logistic before, so Logistic 3.0. Kaboom is for, it's a method we developed in the early 2000s to look for fusion genes. We were looking for correlated breakpoints. So this stands for the correlated analysis of breakpoints, my. So a Kaboom 2.0 project-wide. Yeah, so any suggestions would be helpful. Great, so rearrangements. What about, so that was copy number alterations. What about just the rearrangements themselves? So we see negative selection in rearrangements. Well, for that, we looked at the 2,700 genomes in the pan-cancer analysis of whole genomes, spanning 30 different tumor types and almost 300,000 rearrangements. So these are data that were generated across the world. And we looked at where rearrangements occur both in 1 dimension and 2 dimensions. So this is that 2-dimensional plot that I showed you earlier, but flipped over. And this is the 1-dimensional plot. So sorry, that's odd. So here we're looking at either which points of the genome break the most up top, or which loci are connected to each other most often, which is down below. So we did both of those analyses. The top analysis, one of the most frequent things to come up, or most significant things to come up, were these deletions here, these frequent deletions in the promoter of BRD4, right? So they tend to happen in ovarian cancers and breast cancers, and they often take out the first exon of BRD4, and sometimes subsequent exons, sometimes not. So what's going on there? Is BRD4 a tumor suppressor gene, maybe? Well, in fact, BRD4 is involved in an oncogenic event in BRD nut fusions and nut midline carcinomas. And it's thought to be, if anything, an oncogene. The functions are that it sort of marks DNA that's to be transcribed and then sort of aids in transcription, right? So it tends to localize with acetylated regions of DNA or acetylated histones. Right, and it's actually been proposed as a therapeutic target. It's been used in the clinic because nut midline carcinoma is driven by these BRD4 nut fusions, and so it's been used against that. And then also, transcription factors like MYC, they turn on transcription across much of the genome, and so it's been used to inhibiting BRD4 has been used as a way to target target transcription factors like MYC. Right, so it's a proposed therapeutic target, but we're seeing deletions of BRD4 as being recurring in cancer, right? So I'm saying that cancers don't like knocking out BRD4, inhibiting BRD4, and yet we're seeing these deletions. So what's going on? Well, these deletions are occurring in a context, right, where in almost every case where we see a deletion over here in the stars, it's in the context of larger scale amplifications across this chromosome. This chromosome is chromosome 19. Cyclin E1 is out to the, I forget, left or right. I forget which side. But and cyclin E1 is one of the most frequently amplified genes in the cancer genome, particularly in breast and ovarian cancers. It's a very important oncogene, right? And when it's amplified, we're often seeing these deletions of one copy of BRD4, in the promoter region. So what's that all about? Well, that deletion of that one copy means you're not taking out, the promoter region. It means you're not taking out BRD4 entirely. It has two isoforms, and you're reducing expression of each relative to what it would have been in the amplified state. Right? So here we're looking at breast cancers and ovarian cancers, their expression of either the long isoform or the short isoform in tumors with the focal deletion versus without. And this is their expression after controlling for the number of copies, the amount of expression per copy. And we're seeing that in all but one of these cases, you're seeing significant decreases in expression per copy. So we're still expressing BRD4, but just not as much. And so that's suggesting that it's a toxic gene, right? So cancers want it, they need it. It's a therapeutic target. Well, I don't know if therapeutic, but it's an essential gene for many cancers. But they also can't tolerate too much of it. And so we ended up working on a paper to describe this, and that paper is driven much by Veronica Rendo, who's in Sweden now with her own lab, and the first author was Jeremiah Walla, and this is one of those papers where you write the paper, and when you're almost done with it and about to submit it, you tell the first author, oh, by the way, we wrote a paper, we're your first author, hope you don't mind. And why did we do that? Why did we write the paper without Jeremiah? Well, he actually did a lot of the work, the discovery work. But if you look, he was my graduate student. He's 6'3", and so whenever I stand next to him, I look short. And so of course I'm going to want to stay away from him. So so we sort of avoided him until the very last minute. But now the paper is out actually, just came out in Cell Genomics. And what we showed is that if you overexpress BRD4 in ovarian cancer cells, the cells do in fact grow more slowly. They don't like overexpression, right? If you delete BRD4 in those cells, they also don't like it, and they don't grow as well. So it's sort of a Goldilocks thing. If you overexpress and then in those cells delete the wild, like the endogenous copies, then either the long, so either overexpressing the long isoform or the short isoform. So these are the cells without the guide RNAs. They just have the overexpression. They pop up. So they like having their endogenous copies deleted when there's overexpression around, right? Makes sense. So it's a Goldilocks kind of thing. So BRD4, the conclusion that BRD4 events are preventing overexpression of a toxic gene, right? So we're seeing deletions of essential genes as being toxic, and also as being under negative selection, and then amplifications also possibly undergoing negative selection. So there are other sources of information to identify toxic genes in cancer. So one of them is expression levels, right? So, you can imagine when a chromosome arm undergoes amplification, so that, to amplify an oncogene. Well, amplifying this oncogene leads to advantages in cell growth. But then there are many other genes where the amplification has no impact on cell growth. But there's some genes where the impact would be negative. These are the toxic genes, right? And you would expect that cancers would find a way to prevent their overexpression. So you might expect compensation, that as copy number increases, for these two genes, you would see increasing expression. For this gene, you would not see increasing expression. So we call those Argos genes, for amplification-related gain of sensitivity. And I did not come up with that acronym. But that's one source of information. Another source of information is ORF screens, right? And a lot of folks have done ORF or CRISPR-A screens. So CRISPR-A screens are a good source of information too, which we haven't taken advantage of yet. But we took 18 or 19 ORF screens that other folks have done. So often people do these ORF screens where they take cells and treat them with chemotherapy, and then and then overexpress every cell gene in the genome to see which of those genes will lead to resistance to chemotherapy, right? So they're trying to find ORFs that are positively selected under chemotherapy, and well, they have to have a control, right? Where they don't just want a gene that just helps cells grow better at any time. They want it specifically in the case of the chemotherapy. So they have controls where they don't treat with chemotherapy, right? So we realized that those controls were a great source of information for toxic genes, right? Because you're overexpressing, in these ORF screens were like 10,000 genes. You're overexpressing like 10,000 genes of the genome and seeing their effects on cells across 18 different lines. So, which of them are toxic? So we begged the data off the people who generated these data so that we could be research parasites. And I don't know how many of you know about research parasites, but this is after the worst editorial ever in the New England Journal of Medicine that said people who use other people's published data are research parasites. So we decided to do that. This is the team of folks who did the actual work, including Veronica again, as well as Uri Ben David, a former postdoc of mine who's now at Tel Aviv, and then a couple Flores, who's Foyer, who's a great collaborator in the Netherlands, Maria Kolomitache in Helmholtz, Munich, who's also a great collaborator, and Michael Schubert, who was mentored by both of them and is a great computational biologist. And so we decided to take those data and try to see which what are toxic genes like in the cancer genome? And so we looked at the ORF screens and identified, okay, I guess there's 17 cells, looked at which of the genes was toxic in those experiments. And then we looked at both, well, TCGAs, almost 8,000 tumors, and looked at the effective amplification on expression of each of those genes, as well as the CCLE set of cancer cell lines, 850 cell lines, and looked again at compensation of expression, so which genes expression did not correlate with amplification. And so we looked at and we tried to look for genes in the middle of all those three. And we found a set of genes that were consistently under compensation here, both across cell lines and across TCGA. And these included, actually some genes that you might expect to be toxic, like P21, CDKN1A, which is a known sort of tumor suppressor that when you activate it, it can lead to cell stalling and apoptosis, right? And we looked at the ORF screening data, and we found that out of 12,000, almost 13,000 genes that were assessed in these ORF screens, we found about 589 that appeared to be toxic in at least one tissue type, and it was like 341 that were toxic across all tissue types. And we then looked at the overlap between that. And so these are sort of, bless you. So the genes in the sort of, what was I trying to say? The complexes. So we put these into complexes and asked like which complexes have, appear to be the most toxic. And these complexes up here sort of scored the highest. In particular, and so many of these complexes that scored highly were RNA processing complexes. So the SNRP-free U1A complex, this paraspecial ribonucleoprotein complex, and so on. So spliceosome is another important complex. So all these red ones are significantly enriched. So it appears that overexpressing RNA processing complexes is often not tolerated well by cells. RBM14 was one of the top genes that we identified across all these things. So we went on to ask, like, is its overexpression toxic? And we did find that increasing amounts of overexpression using an inducible system led to decreasing levels of growth. Not massive decreases in some of the lines. This line not so massive. This line not bad. But definitely decreases. So it looks like negative selection. Why is there negative selection going on? Well, we did RNA sequencing and we looked for differences in splicing and everything. We found nothing, right? All that we could find was that there seemed to be poorer repair going on, like more poor DNA damage repair. So we saw here, this is an NHEJ reporter. We found that cells with RBM14 overexpression actually had more activity of NHEJ and less activity of HR, homologous recombination. So NHEJ fixes DNA damage, double-strand DNA breaks by just putting them together, gluing them together, regardless of what their sequences are. HR identifies matching sequences and puts them together. HR has much more fidelity than NHEJ, and so what it seemed is that overexpressing RBM14 sped up NHEJ so that it ended up taking over more of the DNA damage repair, and then you get different, more DNA, more cell death due to misrepair. And so, in fact, when we look at this is, I think, the same thing, sorry. When we look at errors in mitosis, we found that overexpression of RBM14 consistently led to mitotic errors across different cell lines. And you could sort of see those with metaphase bridges, micronuclear formation, and so on, so that the number of mitotic defects was higher. So, RNA processing genes are coming up to the top of the list, but maybe that one of their main functions is in control of DNA damage repair. And this is, with more misrepair, then we start seeing more activation of STING, and the internal immune pathways, innate immune system, and presumably that's leading to cell death. So I think I'm at time. So just to summarize the whole talk, negative selection is infrequent for single nucleotide variants, but no one should care about single nucleotide variants because they don't matter. What matters is copy number alterations, and negative selection is important for that. Understanding negative selection is useful. There's a potential for identifying therapeutic targets, which I haven't gone into, but then also you need it to understand positive selection, and understanding the boundaries of genetic alterations in cancer is a good way to look for both positive and negative selection. And with that, I want to thank the folks in my lab and our collaborators, and all of you. I should mention that Julianne Shi did the BiSKIT work. She started the project when she was entering medical school and at Tufts, and sort of did it on the side in medical school, and then completed it during residency, and it made it into a first author Nature paper. In this fall, she's going to be joining BU as a pulmonary fellow, in clinical fellow in the pulmonary division, and probably will end up doing a postdoc after her first year. So if you can get her, she's awesome. And then all the other folks too. I'm happy to take any questions.
That's really interesting. I have a question about the BRD4 promoter. It looks really important, but why is the conservation so low?
The conservation of the I don't.
Why is the sequence conservation so low in those regions of the promoter of BRD4?
Yeah, I don't. I'm not sure. We didn't look so much at the sequence conservation.
Using Milo P, fast con scores that kind of
across species.
Yeah.
Yeah, it's a great question. I'm not sure.
It looks really important.
Yeah, I mean, you're right, because it's incredibly important in determining tissue-specific expression, right? So it binds to enhancer regions, and H3K4 acetyl regions, and leads to expression of tissue-specific genes. So I imagine it may be that other species, they want different regulation of expression across tissues. I don't know. That's a good question. I hadn't thought about that. Yeah. Yeah?
So, can you apply the same sort of approach to advanced transportations?
Yeah, it's a great question. So we absolutely tried to do that with the 2D plots I was showing you, like the thing that looked like the high C plot. We've developed methods to look for dots where you see connections between regions of the genome more often than you'd expect by chance. And so we've developed those methods, we've applied them, and we identify all the known fusion events that are present in our data. We identified new ones. We identified one new one that validated, but we were scooped with. But there are challenges there, which is that, again, negative selection is shaping that a lot. And so it's not, sometimes it's, well, alterations in chromosome structure are very difficult to understand, partly because of the annotation, right? So if you see a rearrangement, is it its effect because you're bringing together two things, or is it because it's causing copy number alteration somewhere else? Is it, being, are you seeing something connect to something else because it's trying to avoid a third thing? So there can be a lot of different explanations. So we are finding events that are occurring more often than you expect. And so the task now is to try to understand why. Did that address your question? Great. Yeah?
Yeah, really nice talk. Really
Thanks.
interesting. Curious, a lot of the examples that have popped up in these negative selection are these essential genes, important for any cell, right, almost lysosome stuff, maybe the exception of BRD4. But you do the analysis in more like per tumor type, you see more like immune specific or even that particular
Right. So what you're saying is that if you find a gene that's essential to cancer, but then it's also essential to the rest of the person, that would not be a useful therapeutic unless it's somebody you don't like. Yeah. So we're in the business of trying to make cancer treatments for people we don't like. But no, we've done analyses across tissue types, and we definitely do see differences. And another way to say that is, do you see differences across genotypes, right? Like, our P53 mutant tumors, do they have different regions of essentiality than P53 non-mutant tumors, right? And when we do the analysis, when we look for regions in one group and look for regions in another group, we see different regions, but we don't know whether that's just because we're not powered to see some of the events in one group versus the other. So we're developing a method to actually formally ask, if this region is under negative selection in this group, is it also under negative selection into that group? So to look for the synthetic lethality thing.
I guess, I'm sure you would ask about whole genome doubling. Do you see differences like pre and post whole genome doubling?
Right, yes. So we did that analysis because the reviewers asked us to. And not that we care about whole genome doubling. And we did not find a significant difference in the locations under selection, but we did find a difference in the degree of selection, right? So the effects of gains and losses appeared to be blunted in the whole genome doubled cells, which makes sense. Yeah.
What is your take on the regions that are frequent breakers? Like, what's your thought on column fragile sites? I feel like, is there any sort of validity to that in the things you're seeing? Are they homopolymer sequences? What are your thoughts?
Yeah. So I mean, so a lot of folks have looked into fragile sites, and I think what makes them fragile, and I think they do form. You can predict them to some extent based upon the sequence features, like and the likelihood of them forming sort of different structures of DNA. The fragile sites tend to occur in the frequently deleted regions tend to occur in areas that have few genes, again, negative selection. And they tend to be in regions where you have like really massive genes, not genes whose cDNA is massive, genes whose footprint is massive, right? And usually that's because they have a massive first intron. And that first intron, the bigger the intron, the less frequently you express the gene. And so those genes are often expressed primarily in the brain, and they're massive because it's a way of preventing their expression, in other tissues or whatever. So I think those deletions, that all sort of adds up to make me think that those deletions are generally not driving cancer, but they're just happening, and there are sort of mechanistic reasons that it's breakability that's causing them, but also a lack of negative selection. Yeah. Any other? Great.
All right, thank you so much for
Yeah, thank you.
Thank you. That was great. Yeah, really interesting.
I appreciate it. Yeah, well, thanks for giving me the opportunity. Yeah.
Chrissy, nice to meet you.
Yeah, good to meet you too. I guess you're next on my list, I think. Is that right?
Yeah, that's what we were plotting. So Chris is in a different building.
Okay.
And so we were discussing whether it makes sense to go over there and then come, because then you're meeting with me after. So we were just chatting about whether, if you were interested in a coffee, hang out here, or.
Happy either way, whatever you folks.
Whatever works for you.
Did you have like a computer you wanted to show things, or just want to chat?
Probably just in the chat.
Yeah. I can keep going. Maybe choose back end of the L1.
Yeah, okay, so recording.
Yeah.
Okay,
Yeah.
great.
Okay, so.
Okay, so yeah, sorry.
I don't know. Take your time.
Yeah. But yeah. Oh, thank you. Yeah. How is everything going for you?
Oh, good, good.
I've been asking you about administration.